Next, we want to welcome Aimee Dudley from the Pacific Northwest Research Institute. She's going to be talking about improving the diagnosis with the awesome power of yeast genetics. Aimee is a gem and has a ton of energy for this community. She's working to explain those genetic results that you get. When you get a variant that you don't really know what it means, what do you do with that? Aimee's looking for answers to that, and she's working closely with our UCDC physicians to help us understand this and to help us apply meaning to all of these results. And she has committed to studying the genetic variants of all of the urea cycle disorders. She started out with OTC, but we'll be doing all 8 disorders. So thank you, Aimee, and welcome.
Thanks. First of all, I want to say it's a real pleasure to be here. This is the first time that I've attended this meeting, and it's just been lovely to meet you all. Great. So today I'd like to talk to you about some of the work in my lab where we are using the awesome power of yeast genetics, and I'll tell you what that is, hopefully to improve the diagnosis and ultimately treatment of urea cycle disorders. So that's a QR code if you want to scan that and learn more about my lab. First, I'd like to give my disclosures. So my lab does receive sponsored research funding from Moderna. Moderna is a company that is developing therapeutics for OTC deficiency. Moderna did not fund any of the research that I'm going to talk about in this presentation, though. I'm also a scientific advisor for a company that spun out of technology developed in my lab, but that's not relevant for this talk. Okay, so who are we and how did we get into studying urea cycle disorders? So first, my lab is located at the Pacific Northwest Research Institute. We are an independent nonprofit research institute in Seattle, Washington. For those of you who have been to Seattle, you know that we're the home of the Space Needle, Pike Place Market, where the guys throw the fish. It is a lot of fun to watch if you haven't been there and done that. We're the home of Mount Rainier and a couple of national parks, and it's a really lovely place to visit, so I hope you will all have the opportunity to do that at some point. The next is I'd like to talk about kind of what kind of research we do. So when many of you think about medical research, you're probably thinking about these kinds of scientists if you've participated in clinical research studies in the past. And so for those of you who have done that, first I want to say thank you. I want to say thank you to the patients, the families, and the clinicians who are participating in this really important research. And I hope that you'll see kind of as we go along in the talk how even though our research isn't using specifically patient samples and specific interaction with patients, how critically important that is. So thank you. However, I also want to say that we're not those kind of scientists, we're these kind of scientists. So people in my lab, and these are some of the people in my lab who have done this research. Russell Lowe, Russell, come on, stand up. Russell Lowe is a member of my lab who led the OTC study, and he's now leading one of the studies of ASS1 as well that he's currently working on, together with Michelle Tang and Gareth Cromie in the lab. We are all essentially basic scientists, so we work in the lab. Even though we are basic scientists, which generally means kind of, developing, looking at kind of basic science questions and understanding kind of important biological questions, we also do technology development. So both I and Russell actually have for many, many years developed new technologies that can answer questions better, faster, cheaper. And Sandesh was talking about kind of breaking apart these silos. So one of the important things about the study that I'm going to tell you is that this is a very close collaboration with clinicians and disease experts. And so this whole collaboration that has been really powerful started out with a serendipitous introduction to Andrea Grootman. As I think you probably all know, if the Energizer Bunny went to medical school, it would be Andrea. So Andrea, who can never say no to anything, was sitting on a panel with one of my colleagues, Claudia Carvalho, who is a rare disease researcher at PNRI. The two of them got to talking about what they did, and Andrea was talking about her work with urea cycle disorders, and Claudia knew that my lab was developing new technologies that were relevant to urea cycle disorders, and we were actually working on OTC at the time. So Andrea didn't miss a beat for this opportunity for our groups to interact, and she put together a research team for collaboration together with Nagamu, who will be speaking, and I'm sure most of you know, as well as Luba and Hiroki, who are disease domain experts. So they were both postdoctoral fellows in Mendel Tuchman's lab, and they've been studying urea cycles and have made a lot of discoveries about urea cycles over the course of their careers. And so our groups have been meeting for over 3 years now, and we have a call almost every 2 weeks, and as busy as Nick and Andrea are, they often take break from clinic, and we're all on that call every 2 weeks, and it's one of the things that I look forward to twice a month. The other thing I want to tell you before I get right into it is that, as Jill mentioned, I'm going to talk today about OTC, but I want you to know that OTC, I'd like you to think about what we're doing with OTC as the example. And so we are doing similar things and even slightly different things with many of the other urea cycle genes. Andrea and I just had a grant funded looking at all of the enzymes and transporters here that are in pink. Russell has data on ASS1, and Michelle in my lab has data on ASL. So more soon, more to come very soon. Okay, so how can genetics give us faster and more informative diagnoses? Before we get into kind of exactly how we do that, I just want to remind us all a couple of things about genetics. So not all things that make us sick have a genetic cause, right? My husband gets food poisoning, my mom gets the flu. Those aren't the result of genetics. However, for diseases like the urea cycle, disorders that have an underlying genetic cause, those allow us to do special things with them. And so that allows us to do DNA sequencing, and being able to do DNA sequencing, and your experience with DNA sequencing might be something like there's a blood sample taken, DNA is isolated, sequenced, we read out the DNA sequence with the machine, that sequence is analyzed with computers and computer programs, and then that information is returned to physicians who can use that information sometimes to make diagnoses. So what are the applications of that? That has the potential to shorten diagnoses. It has the potential to rapidly diagnose sick newborns, critically ill newborns. It has the potential to expand newborn screening to new diseases that don't have a reliable biochemical assay. And it can provide a non-invasive prenatal test. So one can actually sequence the baby's DNA from a sample of mom's blood, kind of late in pregnancy. And so there's a lot of potential here. But before we talk about the potential and the caveat, we just need to know a couple of things about DNA. So when I think of all the DNA that we have in our cells, right, we call that the genome, and that genome is a long string of the G, A, T, C letters. So it's about 6 billion of those letters. And we can read those letters from start to finish now. And I like to think of what this DNA does as a book. It's an instruction manual, if you will, that tells a cell how to build and operate a human being. And Oh, I'm so sorry, my slides are screwed up. Oh, this is all going to be a mess. And within, so for that book, we can think about these DNA sequences as a sentence in that book, right? So a gene, like OTC, for example, the gene that encodes OTC, is a sentence in that book. And you can think about DNA sequencing as reading out that sentence in the book. And when we have the version of the gene that most people have, sometimes people call that the wild type sequence, that sequence it performs some function, right? So OTC performs some function. And so the version of that sequence that most of us have are a wild type sequence. And so that might be a sentence that we might read across that would be something like, you know, a cat sat on my pillow, if we want to convert that G, A, T, and C into something that is more kind of human readable, okay? And so, oh, this is all going to be messed up. So if you have a version of the DNA sequence that has a change in it, not all changes actually cause disease, right? And so sometimes you can have a sequence that goes from one version to another version, and that sequence change in your genome, that spelling change, doesn't actually change the meaning of the sentence. It doesn't actually change the function of the enzyme, and people call those benign mutations, okay? And so you might change it from a cat sat on the pillow to the cat sat on the pillow. And the cell's interpretation of that doesn't really change, and in this case, Dolly's interpretation of how you would actually draw out that sentence, the cat sat on the pillow and a cat sat on the pillow, they're essentially the same thing. But sometimes you have a sequence change that really changes the sequence of the gene, and so you know, a cat sat on the pillow and a rat sat on the pillow, those are two very different things. And when those sequence changes cause disease, those are called pathogenic variants. However, for the vast majority of DNA changes that we find in people's genomes, and there are a lot of them, we don't know whether or not they're likely to cause disease or whether they're likely to be harmless. And those are called variants of uncertain significance. And those are the bane of everyone's existence, because you can't, when you don't know the functional consequence of a DNA sequence change, you can't use it for diagnosis and you can't use it to treat patients. And so these are frustrating for geneticists and physicians. They're confusing and anxiety-provoking for patients, and we need to do something about them. Okay, so the good news is that you don't need to remember any of those terms. There isn't going to be a pop quiz, and you just need to know really what's important here. And so what's important is that some DNA changes are harmless, some DNA changes can cause disease, and for most of them, we just don't know which it is. And so the scale of this problem is enormous. So there are 60 million potential types of DNA sequence changes of the kind that my lab studies that could occur in the human population. They probably do occur in the human population in some people somewhere across the world. And of those 60 million possible, 59.9 million of them are currently variants of uncertain significance. We have no idea whether or not they're harmful or harmless. Okay, and so I can show you a graph and some data that talks about all of that, but I really like this one quote from the literature, which I think sums it up really best, which is, my, but you do have a problem, a big problem. And so this is a really big problem in the field of human genetics. Okay, so now we can talk about urea cycle disorders. So for OTC, what Russell wanted to know when he started his experiment was which of the 2,000 possible OTC variants are likely to cause disease and which are not. And so we need to be able to functionally test each and every one of these. And we can test them all, but we can't do it in human patients, and we can't even do it efficiently in human cells. So enter the awesome power of yeast genetics. So yeast is a powerful model organism. So there are thousands of researchers across the world who actually study yeast, and there's a good reason for it. So first, what do I mean by yeast? What kind of yeast am I talking about? There are lots of kinds of yeast. You might have heard about things like yeast infections. That's a different kind of yeast. The kind of yeast I'm talking about and that my lab uses is baker's yeast. So this is the Fleischmann's yeast that you buy in the grocery store. We all got familiar with this during the pandemic, making bread. It's the kind of yeast that's used in making beer and making wine. It's also, my lab has done some studies examining the yeasts that are involved in the production of coffee and chocolate. And so in addition to its value as an important organism that makes beverages and foods that many of us enjoy, it is also a powerful model system because we can do experiments faster, cheaper, and much more easily than we can do in other systems like mouse and humans. But many biological processes that are important for human cells also perform the same functions, also happen inside a yeast cell. And because of that, we can actually use yeast to study processes that are important for humans, and those processes include the urea cycle. So how did Russell test almost 2,000 variants that are in OTC? So I'm not going to go into the details. We've published a paper. We have a really nice article written by Jill that describes in a non-scientific way how this research was done. That's on the NUCDF website, so I encourage you to take a look at that if you'd like to understand more about the details. For today's talk, what I'd like you to know is that the first important thing is that we use synthetic DNA. So you can buy DNA, and you can buy different versions of DNA from companies. We buy it from Twist Biosciences. And the important, I think the valuable thing for that, for you all to know, is that the DNA we're working with, the DNA Russell's working with, doesn't come from patients. It's informed by patient information, but we buy this from a company. We don't take liver biopsies. We don't take blood samples. That's a good thing, right? So Russell orders every possible variant that he's interested in, and he ordered 2,000, and he recovered almost 1,600 of them from a company. He then takes them and puts them into yeast, and he can do this in a matter of days. So he can look at thousands and thousands of variants in a matter of days. He puts them into yeast, and then he isolates out the yeast that contain each yeast cell contains an individual variant that is a variant that could occur in a human and in many cases have occurred in humans. And he then measures how well that OTC variant works. And he does that by measuring, because the way he set up the yeast cells, how fast they grow depends on how well their OTC enzyme is working, okay? So the cells that are growing fast, those have really completely functional OTC. And the cells that aren't growing at all, they have completely non-functional OTC. And then there are cells that are growing somewhere in the middle, and those have a range of activities. So some of them are growing 5% well, some of them are growing 10% well, some of them are growing 100% well. So that yeast growth is telling Russell how well that OTC enzyme is or isn't working. All right, so he takes all of that data, and then he says, okay, I'm going to look at the variants that don't allow the cells to grow at all. These cells are not growing. They're growing less than 5%. There's almost no activity, little or no activity. And then I'm going to look at the ones that are in the middle, like 5 to 90%. So some of these are still pretty severely impaired, and some of them are, pretty close to wild type. Those ones that are up to 90%, those are pretty close to what we call wild type. And then there are ones that are really just growing just as well as the completely functional version of OTC. Okay, so now let's look at how well the yeast assay compares with what is seen in humans. Okay, so Russell started by looking at 69, so the yeast results for 69 variants that had been seen in neonatal male presentation patients. And so the first important observation is that 70% of those variants occurred in the yeast assay. They had a yeast assay result of almost 0, so in that really low activity. So this suggests that that the variants that have almost no activity in our assay are associated with the severest forms of the disease, okay? And then in the kind of still low to middle range of the assay, there were 29% of those variants were associated with late onset, with neonatal presentation. So a smaller number of them were actually in late onset, in neonatal presentation. And almost none of them that had near wild type activity, that had completely functional activity, were associated with neonatal male presentation. So now, I'm sorry, the slides got all screwed up. They were so good on my computer. So now let's look at late male presentation. So now, where are the vast majority of the variants that are associated with late male presentation? And that is, so 75% of those, the vast majority, are now associated in this low to mid-range activity, okay? And again, very few of them that are associated with late onset male presentation are in that high level of activity. And then in female presentation, again, the vast majority of them are associated with really, really low activity. And so kind of what does this say about a person's genetic risk? Okay, so the first thing that it says is that if someone sees a patient or an individual, it doesn't have to be a patient, it could be just somebody out in the population, with a variant that is associated with this very high level, 90% or above, then we don't expect to see disease in males or females with those high level variants. What happens if we see individuals who have this kind of low to medium range? So that's consistent with a disease that might show up in males as a late onset male phenotype. And female presentation is possible, but maybe it's less likely. And then the final observation is that for those variants that have almost no activity, this is consistent with neonatal onset disease in males and with female presentation. So there's a very important point here, which is this kind of functional information is very valuable. It's valuable for research, and it's valuable in combination with a lot of other parameters for diagnosis and treatment. But this functional information is only one piece of information that doctors, patients, and their families can use to make decisions. So that's, if you take nothing home from this talk today, I hope that you'll take home this really important message. And then just one final point. So we talked about kind of where the variants that in Russell's study, the ones that had been seen previously in patients and that were previously characterized through clinical research and clinical studies, we talked about where they were with respect to severity. But another important point is that the vast majority of what Russell looked at, 1,356 of those variants, we don't have any patient data on them. And a really large number of them, 430 of them, are in that incredibly dangerously low range of the assay. And so these are variants that could show up in a human patient, and they are consistent with what we would expect to be really severe forms of the disease, so neonatal onset male disease and female presentation. So what does this mean? So what that last result means is that the variants that, variants that are most difficult for geneticists to do anything with and for clinicians to do anything with are the ones that no one has seen before. And this type of functional information provides some information that we didn't have before about these variants. There are a lot of them, and a huge number of them have potential to cause the most severe forms of the disease. And so the most important part about all of this is that you all are the most important part about all of this. So all the work that's happening here at the National Cycle Disorder Foundation, and the work that many of you are carrying on, the work that Cindy championed so well, and that's continuing, is the most important part. So with that, I would like to thank all of you for your attention. I'd like to thank the families and patients and clinicians who participate in medical research, the members of my lab, and our funding sources.
Thanks, Amy. We do have a few minutes left for questions. So if anyone has a question for Amy, all right, lots of questions here right up front. Here you go.
So, I've been waiting my whole life for this. I actually have my mutation, and I want to know what it means.
Mm-hmm. And so a great way to get the answer to that question is to talk to your physician. And our information is actually on OTC, is published.
Yes.
And so all of that information is public, and so your physician can themselves, or in collaboration, in consultation with all the great doctors here, including Dr. Grotman and Dr. Amu, they can all help you integrate that information to the extent that we have it. Now, we don't always have information on every single one of them.
Yeah. So
Yeah,
I actually
so they can absolutely help you with that.
and I actually got my genetic mutation found. It was very, very complex, and I found it through pregnancy. And I didn't find it until like 2009. So I didn't know what kind of thing I had my whole life. And they used this when I was pregnant also. This was super valuable during pregnancy because they knew exactly where to go to look.
Yeah, so the value of understanding kind of what mutations mom or dad might carry during pregnancy can be incredibly valuable because one way in which that can be used is, I was talking about how it's actually possible to now non-invasively sample the baby's DNA from mom's blood. So amniocentesis might be something that people are aware of in terms of being able to sample the baby's DNA in certain ways, but that, of course, requires an amnio. And so this ability to sample DNA from it's not completely non-invasive because, of course, you have to take a blood sample from mom, but, when we're pregnant, everybody's poking us all the time anyways. So it can be incredibly valuable in terms of helping physicians know whether or not that fetus is actually carrying a particular variant that mom or dad might have had.
Amy's also going to be here for the panel at the end of our next few speakers, so there'll be another opportunity to ask questions.
We would like to know how we could do the test about to measure the severity of the disease. And we have a kid with ASA, and I heard that you are still developing the profile for ASA. Is it possible for us to participate? Is for a kid with the disease still to participate in the study?
Yes, we do actually have a data set that we are bringing through its last amount of quality control. Then a very important aspect of analyzing that data is that we write up the papers, and those go through peer review. So they are rigorously looked at by other scientists and clinicians, and those clinicians and scientists look at our data and look at what we've said about our data, how we've interpreted our data, to make sure that there aren't any concerns and to make good suggestions sometimes about how we can revise the analysis or revise our interpretations, and they usually make really good suggestions. So one of the important things about our research is that those values that we have, And this is actually a really great question, so I'm glad you brought it up. These values that we have, the yeast values are completely independent of what is happening in a human, and that can be really valuable. It can also have caveats to it. But even though, so these values will not change over time, but our understanding of how they relate to the human disease can change over time. So for example, Andrea was talking about how, we're learning more about how females, for example, with OTC variants might have phenotypes that we didn't initially call disease, but we now maybe in the future understand that they're having certain kinds of symptoms as well. All of that can be informed by the yeast without actually needing to redo those yeast experiments. So we use the clinical interpretation on top of that. And so, for example, how one might use this in terms of the ASA study is that, and in fact, Russell is the one who's actually doing that study as well. He is going to have a yeast data set. It is going to say, we expect, these particular variants to have this amount of activity. Is that correlated with severe disease? Is that correlated with individuals having one type of phenotype or a different kind of phenotype? And so all that information is available for clinical researchers to be able to help inform their studies and help inform the clinical studies. So that's a long way of saying that although the clinical data and your participation in these clinical studies is critically important, from our perspective as doing the technology and making that available, there's nothing about any particular variance activity that is going to necessarily change how we do those experiments and how we put out that data to make it available.
Yeah, thank you, Aimee, very much. Yeah, there's a process that they go through with the physicians, and then their interpretations will ultimately be sent to the American College of Medical Genetics, and that's when things will become officially available for use in medical practice. So it is a bit of a journey, but Aimee's work is really accelerating things.
And Aimee's research is in all of your packets. So all of your folders, we included her research there, so you have a copy of it. And then there's a great YouTube interview that Jill and Aimee did together that's all over our social media and also on our website, so if you're interested in that.